Okay, well, I think that we'll get started. Good afternoon. I think it is for most people. Good morning to those people from the United States who are tuning in. My name is Chris Griffiths. I'm immediate past president of the ESDR, and welcome to this week's ESDR Kitchen webinar. Those of you who have been with us since last July will know that we started these webinars back then when we were deep in the COVID lockdown. And there were no face-to-face meetings, pretty much the same as we are now. And we felt it important for us to remain in contact with our ESDR members for educational and also partially social reasons. And we came up with these ESDR Kitchen webinars, which are along 4 themes or 4 tracks, if you like. The recipe book, which is new research techniques explained, the ESDR Kitchen. And that's what we're going to hear about today. Sweet and sour, where 2 protagonists debate a hot topic in the ESDR Kitchen. So we're taking different points of view about a particular important scientific discussion in dermatological research. Freshly baked is a couple of recently published high-ranking publications. We usually have the first authors to take us through those papers. And then molecular cuisine is when a distinguished skin biologist, clinical academic will describe his or her paths of discovery in their career. We go to the next slide, but today we are with the recipe book. This is supported by Almirall and Sonkoly, and I'm delighted to have a previous ESDR board member join us, Lionel Larue, and he's going to talk about biological models to study melanoma, but I'll hand over to my colleague on the board, Julien Seneschal from Bordeaux, to introduce Lionel.
Thank you very much, Chris. Hi, everyone. So I'm Julien Seneschal from Bordeaux, and I'm here with my colleague, Enikö Sonkoly from Stockholm, to share this session. So just to give a brief introduction of Lionel Larue, our guest speaker today. So Lionel has a long-standing research interest in pigmentation and melanoma, and Lionel performed his PhD at Gustave Roussy Institute and then 2 postdoctoral trainings in United States and Germany, working on differentiation and transformation of melanocytes. And now Lionel is a research director at Institut Curie, located in Orsay, close to Paris. And his laboratory focuses on molecular genetics involving the melanocyte lineage with better identification of the molecular and cellular mechanisms occurring during the establishment of the melanocyte lineage, homeostasis, and transformation. So his lab generated several models and approaches for advances in the understanding melanoma and progression. He published outstanding research in high-ranking journals and received several prizes for research in melanoma. He was a former board member of the ESDR and former president of the European Society for Pigment Cell Research. So today in this recipe book episode of the ESDR Kitchen, Lionel will give a talk on biological model to study melanoma, and this is a great pleasure to welcome here in this session. Thank you very much, Lionel.
Thank you, Julien. Thank you, Chris. And thank you to the ESDR board and the fellows which are in Geneva to take care of this, which I think is very important to take care of. So, I mean, today I will speak of biological models to study melanoma. And before to tell you this, speak of this, I just want to tell you that it's not really new research. I mean, it's the fact that we try to make some biological models is very old, because we always try to simplify a question to a model. And these models, by definition, will not represent all the aspect of a problem, whatever the problem is. The second point that I want to make before starting is that melanoma is not only a unique disease, but you have several diseases. And according to the location, for instance, or eventually the origin of these melanocytes in embryological terms. So we must not forget that when we have a model that can be used for melanoma study, we have not to forget that this model is limited to some of this melanoma. Of course, we have some common issues for the different melanomas, but we must not forget that it will not represent all the melanomas. So, saying this, the first thing that I want just to remind everyone is, why do we want to generate biological models in a very specific manner? First, it's of course to understand the function of a gene during development, embryonic development, and homeostasis. To improve or to modify some phenotypic characteristic, such as we know what is going on and how we can play with that. To discover novel physiological or physiopathological functions. To understand the gene-gene interaction using in vivo model. But you can also do some gene-protein interaction at the same time using this in vivo model. For instance, what we can think of is some chip cycle experiment that can be done directly in vivo using these models. We can also evaluate the in vivo consequences of the modification of a gene on a specific lineage. We can now, at the level of tumor genesis in general, not only in melanoma, of course, is to evaluate the role of genes involved in tumor genesis. And tumor genesis, as you know, is not a yes or no. It's a multi-step event, and then sometimes you need some events have to occur before some others for certain logic, and at the level of the cellular level, but also at the molecular level. You have also to identify, and you can identify some tumor-relevant genes, really relevant genes. And then I will not speak of drivers or not. To generate physiological model for models for humans, that we spoke of that, and how we can use these models to have some better ideas how to move on. Of course, at the end is to help discovering novel therapies, but we also must not forget that we can induce a better prevention using these models. When we think of all the people which are working on a field, of course, you have the medics, and then for the medics, you have many medical characteristics, the oncologists, the surgeons, the anatomopathologists, which have their characteristics and strength. But also you have some biologists, which are not MDs, we have physicists, mathematicians, and so forth. All these people, by definition, are different, and their mind is set up in a different way. Moreover, with their education, they have a different angles of apprehending some issues. So we have to see this as a power and not a weakness. It can be a weakness when the people don't realize that it's a strength. And then whatever the people do, first they observe, at least it's what they should do. And then from this observation, they can make correlation. And this correlation is kind of important because you can say, oh, this is often going through this. But you have to be very careful with the correlation because sometimes these correlations are fully wrong. And it's you observe this and this, but they are not related. That's why you have to see the implication and the reciprocity. Of course, in the case of a disease, you have to interpret. And this interpretation, you can go to the diagnostic. And then after that, of course, you don't want to leave this person dying, you want to act. And then you can act in any way, or you can go for the rational therapies, and that's which is different from the empiric way it was going on in the past, and we're still going on. And with some success, we have to admit, but the rational therapy is also more satisfying for our mind. And of course, the action, you have to evaluate the benefits versus the risk. With all these issues, we have to ask, what is the disease? How can we fix it? How does it work? How can we simplify the problem? And then with the right level of simplification. So basically, with the correlation, the clinical analysis is still very, very important, and all the exams which are associated. The anatomopathology is also very important to know what's going on, the biochemistry. And more recently, of course, we have access to imaging, and that is crucial, the molecular histology, and of course, these days, the omics. The omics brings a lot of data. And then when we take a look on the data which are brought into a system, then at that moment you can use the artificial intelligence. And that is something which is fantastic when you are a car maker, a plane maker, or whatever, because it's kind of simple, even though when you take a look on this aspect, it's very, very complex. When we speak of the body or an organism, it's become, of course, very, very complex. And I think we have to work in this direction, but we must not forget that we are still very, very naive. When we think of the omics, we have all these omics. We have the genomics, the epigenomics, the transcriptomics, the meionomics, the metabolomics, the histology, molecular histology that's still useful, and any omics that you want. And that is kind of important because we have a huge amount of information, which are more or less solid, more or less clean. And also we must not forget that for melanoma at least, that you have the melanoma, but you have also some skin which is around when it's not fully dissected, and I'm not speaking of single cell right now. So basically, all these things just generate correlation again. And that's this correlation, you can go to these type of things. You can see, for instance, when you compare different melanoma, then you have the mutation, but mutation is one thing and the genetic is very important, but the genetic is not everything. You have to also to think of the levels of expression activity, not only of the RNA, but also the protein. And that's also something that will be very important to understand. Also for melanoma, there's an issue which must not forget is that the plasticity. What I mean by plasticity is the same cells or descendant can be something, but they will not always stay the same way. Not only at the genetic level, but also at the level of allogenetic, meaning other than genetics. And that is very important because according to the microenvironment or even the environment, then at that moment the cells may evolve in one way or another, come back to what it was before or differently and so forth and so on. So we must not forget that we have to think in 4D as well. So now we have the patient, and then here you may recognize some, not former president, but some guys who wanted to run for US. I mean, I think it's to present a US president is good after the one we had before, and it's not associated to Europe, whatever. So as you can imagine, then you have the fellow, you have his tumors, and also you have all the different aspects that we can see. So the cell in culture, the ex vivo or 3D organization and so forth. So, and then I come back to what I started with was what is a clinician perspective and what is a scientist perspective. And as you can see here is they are very different. And it's logical because it's not the same job. In one case, the clinician, he wants to be able to make the good diagnostic for the patient. He wants to evaluate the prognosis and also, of course, to propose the most adapted therapy to the patient. The scientist is far away from the patient and the pain of these guys. What they want to understand is to understand the physiopathological and histological events, to decipher the cellular events, or to decrypt the molecular events. And that, of course, that we can say that's totally different. In a way, yes, but in another way, no. It's fully complementary, and we must use, again, this weakness as a strength. So what I will speak of is the different models for melanoma. Speaking of the relevance of these models, and also at the end, the alternative method 3R to replace, reduce, and refine. And that naturally you see, and you will see that it's something that the people which are using the model, when I say model, a physiological model, they do think, of course, of this replacement, reduction, and refinement. So first, the models for melanoma. The first models for any human tumors emerged a long time ago. The issue was to establish cell in culture, and then at that moment, to see what was going on, are we able to do it, and so forth and so on. So basically, in humans, you have the initiation and progression, which occurs, And then these tumors were taken, and after surgery, these tissues were established in culture to make some tumor cell lines, or engraft directly in mice to make the PDX that are kind of fashionable these days. And then from this PDX, you can also establish this cell line in culture. Do we have exactly the same characteristic than the TCL? Not always. And in any case, when you take all these cells, you may graft them either in NSG or onion mice. And of course, these mice have to be immunodeficient for obvious reasons. So that's what you can do. And then from that, you can start to do the modification assays. How you can do this, you have these cell lines, so you can include or add whatever you want to these cell lines. You can put some DNA, put some protein, to put whatever you want on these cell lines to see what is the consequence on 2D. But you can also take these cells and engraft them into mice in xenograft various ways, and then according to the way you engraft them, you will have different answers. You can do 3Ds as a tumor spheres, and that it's whatever it is. And you can also put them in organoculture or pseudo-organoculture with some keratinocytes, with some fibroblasts. You can do that in 2D, you can do this in 3D, more sophisticated way or whatever. And then you can also play with the PDX by having a short-term in vitro aspect of this PDX. So in any case, what I just proposed to you was that from all this experiment, we have a lack of information on melanoma initiation. Of course, because this melanoma initiation occurred in the patient. And very often the first step of progression also occurred because we have plenty of melanoma which are able to be established in culture, not at the level of the RGP, but very often at the VGP, which is a late event. Also, what you can think of, and I want to mention that at this level, when you establish melanocyte in culture and non-transformed melanocytes, you have to put this melanocyte in very specific medium, which are different from the medium in culture that with the melanoma. So when you compare the melanocytes with the melanoma, very often people make the big mistake to compare them directly, which is sometimes fine, but sometimes is not fine, because for instance, I would just take one very simple example, is that you need some fibroblast or the TPA, and you put this TPA on the cells. The TPA is acting on PKC. PKC is required to be induced to allow the proliferation of these melanocytes. So from that, you already have a difference from the medium which are on melanoma. Very often they don't need PKC, or they use something else, and that becomes the problem. The second problem is that we know how the immune system is important, and then in immune mice or NSG or whatever, you don't have this immune system which is there. Therefore, you can't have this impact of the pressure of the immune system. So all these models that I presented to you have, of course, their quality, but something is lacking. So from that, we have to find a proper organism to be able to do this. Is this proper organism is there, or to mimic fantastically a human? The answer is, of course, no. So we have in the nature some models which are generating some melanoma. We have fish, like the Griffiths. It's a mutation, for instance, in the EGFR from the Griffiths, and that induces a melanoma. And in mammals, there's 3 good examples, which are the horse, the dog, and the pig, or the mini pig. These melanoma are different. For instance, the horses, they have some skin melanoma and some mucosal melanoma. But it's not very dangerous for the horse. For the dog, they also have mucosal melanoma. But the consequence is totally different. It's very, very aggressive. So basically, from the same type of tumor, in one case, it kills the dog, but it doesn't kill the horse. And for the pig, basically it's an embryonic development problem because these melanoma are already present at birth, and very often they are reduced and disappear with time. And the pig may not die of it. So what can we do with this animal? We can do the genetics as we do for humans, but doing experimental manipulation of this animal is kind of complex for various reasons that I will not go through. However, we have two animals which are able, and we know how to modulate and affect their genome. The mouse is kind of easy, and zebrafish. None of these animals are able to form naturally some tumors, some melanoma. It is important to note. So that is the first pitfall if we think of that. For instance, I will just take the example of the B16 melanoma, which is a melanoma which was established a very, very long time ago. And the guy who did that just put basically whatever he could as a carcinogen to get these tumors. And I can tell you, when you go back to the first papers associated with B16 melanoma, you just say, what is going on with this B16 melanoma? And when you take a look on the genome of these cells, they are very, very, very different from what is going on, and nothing to do with what we can see in humans. So basically, with the mouse and zebrafish, you can manipulate them. So let's take a brief look at what is going on in the patient. So here that it's a general scheme, very simple. The epidermis, the basal, where you have some melanocytes, and there you have some nevi. We can have access to that, and we can have access to all these melanoma. What is going on really in the back of that? You have the normal skin, you have the nevis, atypical nevis, RGP hyperplasia, VGP, dissemination, and so forth and so on, and metastasis. During all these stages and phases, you have different cellular events. You have proliferation and oncogene-induced senescence, the bypass of senescence later on, then at that point, the proliferation associated with a pseudo-EMT and invasion, and then after that, you have the dissemination associated with metastasis. The scheme that I'm presenting to you is, of course, wrong. It represents a very low percentage of what's going on, and so forth. The issue is not there. The issue is that you see that we have very often in culture these melanoma from the humans. We may have these cells in culture with some very specific media, and that it's also rendered difficult the game. So what we know so far with all the experiment that was done with mouse, then the MAP kinase and also humans, the MAP kinase pathway is very important for the first step for the initial proliferation. NF1 can be mutated, NRAS can be mutated mainly in Q61 and not in 12, as in many other type of tumors, and that is also interesting. And then we can speak of that, and it's certainly associated with the interaction with very specific protein which are present or not in melanocytes compared to the other cells. Of course, the BRAF, you know that. For the bypass of senescence, either you go through the RB pathway, or either you go through the P53 pathway, and so forth and so on. So with this mouse, what I want to tell you that we could learn a lot of things on the initiation that we do not know of in humans. For instance, I mean, I will give you some example from the lab, which is fine. It's, for instance, with individual tumors, you can see that depending on the status of your mice, which are mutated for BRAF, BRAF P10, BRAF P10, BRAF2, and so forth, then at that point, you can have an information on the number of individual tumors, which means, according to the background, you may have more initiation. you may follow the growth rate, which is totally associated with the proliferation. You can see the appearance of the first tumors and so forth. And you can also see when you have some metastasis, here the metastases which are presented in the lung, for instance. So you can have all this information, and then after that, you can do whatever you want at the level of the readout. So basically, what we have to appreciate for the transgenic technology, in 74, that was the first transgenic mice, and that was with the virus. Then in 80, you had the first transgenic mice with polyclonal injection, 89 knockout, 94, the conditional knockout, and then 2002, it was the end. So basically now we can do the CRISPR and so forth and so on, and with the advantage and disadvantage. The first model which arose and was produced for melanoma was in 91. A very basic model with the equality and so forth. And then it was all the others who went on with a different technology. So here I give you a table which is associated with a different melanoma that was generated. And then basically it can be cell autonomous as monogenic or SOPs, or it can be also associated with carcinogen or not. There was melanoma, there was some metastasis or not, and so forth and so on. So basically you have all these things. That's monogenic, they can do it or they can't do it and so forth. You can have also multigenic, then at that point you need more than one gene, which is important, and then they give more effect, or you affect more, the initiation is faster, or the formation of metastasis is more important, and so forth and so on. All of these have different characteristics. And there is also something that we have to take into account, that is the non-cell autonomous. So basically you have, when the keratinocytes are mutated, and only keratinocytes which are mutated, they may induce some melanoma by action of the exteriors for the different production, or in this case, in the absence of the RXR alpha or the absence of TAF4. That you do induce melanoma only after carcinogenic action. But that we will see in the future, that also very, very important for the initiation of melanoma in certain contexts. So basically what we can do with the establishment of mouse tumors, and I will not go into details how we do these models. It's technically, I thought it was not the point to do that here. What we have a mouse which is healthy. Then at that point you induce initiation and progression, and then the mouse becomes sick. So what you can do, you make the surgery, then either you take the biopsy, and then basically you reimplant that into the mouse. Either you reimplant that in orthotopic or onto the fat pad or wherever you want. And then you have these models, which we can call that GDI. And then you can, of course, establish melanoma in culture. You can establish them, the myocyte cell line directly, or you go through a nude mice. You can also do this. An important issue, when you do this type of experiments, when you establish a melanoma either in congenic mice or in immunodeficient mice, very often you see that the chance of success is much higher when you go to a congenic mouse. And the reason is certainly due to the fact that you have much more natural killers in the nude mice, for instance. So just an apartheid. So now, for instance, you can have some fully coherent model, and that it's an example with the NRAS. We have the tyrosine NRAS mice, and then these mice are growing, and after 1 year, you can see that you have one tumor. So with this, you can establish the normal myocytes, which remain non-transformed, and they're perfectly normal as far as we can say. But the fact that they have NRAS in culture, but they don't. When you reimplant them into mice, they don't grow. when you remove the TP I was speaking about, they don't grow, and so forth and so on. Now, you can also have these tumors. You can freeze them, you can inject them into mice, or you can establish some melanoma in culture from them. And then these cells, of course, you can form some tumors or whatever you want. So the advantage of that is that you have some cells which are mutated for one gene, and some tumors which are mutated for that same gene, but many events went through this. And then you can compare and see what was all the events who had to occur in vivo to become a melanoma. And then you can test that on the cells to see, is it the cells or is it the molecular event that you need? So now you have an animal which is healthy, and if everything is okay, it will remain healthy. Now, when you treat them physically or chemically, and they have some genetic issues, then at that moment they can generate some melanoma. And that it happens in many, many cases. Nothing is happening. You know, you can make many mutations to the animal in the melanocytes specifically. Nothing will happen. They will remain perfectly normal. But if you add something else, then at that moment they can move on. So it's exactly the same thing that when you do a second mutation or third mutation and so forth and so on. So basically here the same thing. Then of course you have all this melanoma, and then you can do targeted therapy. You can do immunotherapy, and you can do also targeted radionuclide therapy, for instance. This immunotherapy, we can do it when we are in a black 6 mouse, for instance, or in a non-immunodeficient mouse. But you can also play, of course, with cell lines. So an issue which is also important is the relevance of these models. And that is kind of simple. We have some information at the molecular level from the humans, and then we want to test what is the consequence at the molecular level. So basically what you can do, you do this modification in this mouse or zebrafish if you want, and then you just follow the physiology and physiopathology. Then from that, after that, you can do the clinical analysis that you want, and then you have access to the tissue. Then after that, you can have access to molecular histology, and then you can have the cells, and you can do biochemistry and cell biology. And then each time, you have to wonder, what would be the best for each thing? At the molecular genetics, you have to put the exact same mutation. But then you have to wonder, okay, do the exact same mutation, but the gene in mouse is different from human. For instance, what you have to appreciate that for BRAF, it's just a non-important point, but I just want to remind you this. In mouse, the BRAF V6 order, it cannot exist. It's V599. So there's one amino acid less than in humans. So the exact same mutation cannot exist as such. However, the function is similar, and we can play with that. So you have to think of that at the same time. The dose also is important. Imagine that in humans, you have the dose, which is, let's say, 1, whatever the dose is, 1. And then you want to make a model, and you put 1,000 of that gene. Of course, you may have an effect, but would it be relevant because you put 1,000 times more than what you should? The time is also important. If you induce the mutation during embryologic development that is not occurring in humans, it occurs just when they are 20 or 30 years, then at that moment, it's totally different for many reasons. Of course, the mice do not live for 20, 30 years, but then with the chronobiology, you can play with that. thankfully, with all the technology, we can do a lot of clinical analysis, and also the in vivo imaging is very, very important. The molecular histology that you understood already what we can do. We can do all evaluate the dermis epidermis localization, the level of differentiation, heterogeneity, but you can do all the omics that you want at this level as well. And that is also very, very nice to do. And then when you establish the cell in culture, you can do all the experiment that you want at the same time. The advantage here, when you have the mouse, when you have the melanoma cells, then at that point, you're coherent at least on several issues, which is that you cannot do that very often with the humans. And then each time, what you have to think of about your model, according to the melanoma of interest, how good you are for that. And then when people ask you, what is the good model to study melanoma? There is no good model. There is some model to eventually answer some specific question, of course. And then each time you have to wonder for each aspect of it, how good it is. And then you have kind of rough idea of what is going on. So now, when we take a look on all what I just said, that's basically this little scheme. When you have the humans, when you have the cell lines, when you have all the omics that we have with histology, and all these things go to these computers where you'd make as much as analysis as you want. When you want to make the link between the human and mouse, you can do that through the molecules, as we can do with the transgenesis, or you can play with the mouse to get some more information, to have some pseudo 3D or physiopathological issues. And then, of course, at the end, when you have the molecules, you can compare this. Then from that, maybe one day we'll be able to play better with the AI, but so forth, it's far away to be simple. And also, as I said at the beginning, we have to move from the correlation to the implication and reciprocity. And the only thing I want to finish with, it was to speak of the replacement, reduction, and refinement. And that it's, as I said, it's kind of obvious that naturally all of us do these kind of things just because we are dealing with, we try to do as less as possible of experiment in mice or whatever animals, because it's long, because it's expensive. And then why we want to reduce it, just because, again, it's a question of money, but also because we can do it now with all the in vivo imaging. And refining, of course, we want to refine this because our questions are becoming more and more sophisticated. And then just to finish, this model may help to induce a better prevention, and I think that I want to insist on that, to assess the processes of transformation and to discover novel treatment therapies. And I would like to thank you all for this, and I'm ready to take all the questions you want. I want it to be very, very large, and didn't go into details in something that because I'm sure that you had the chance to follow many talks, but
yeah. Thank you very much. Oh, there is some problem with the sound. Thank you very much, Lionel, for this beautiful overview and showing us all the different aspects of biological models of melanoma, different models. We just have time for one very brief question, and we have already one question here in the chat. So you showed these tumors in the horse and dog, fish and mini pig, and the question is, are these the result of the same genetic alterations? And if so, do you know why these tumors are so different?
Okay, so I guess one question, okay, for the Griffiths, as I said, it's the equivalent of EGFR. For the interesting question to me is associated with the dogs and horses. Why they have both mucosal melanoma, one is very aggressive and the other is not. And it's a question that I'm asking for a long time to myself. We don't really know what are the exact cause of this mutation, the exact cause of this disease, sorry. But there's one thing that we may think of. And and it's about the fact that horses are basically they are vegetarians, and dogs are mainly carnivores. And then when we think of the microbiota and the importance of the microbiota associated with the food, then at that point it would be of interest to do this. So now and then I look for this, do we have the information? For instance, in India? And compared to the Inuit, which are eating only some food and so forth, and it's just very basic things, do we have information on that? And I could not find anything about that, just for the environment. Of course, you will tell me an Inuit and a person living in India, which are vegetarian, has not exactly the same genetic patrimoine, which we agree, but that would be something of interest to me to try to go much more on this direction, to try to understand what is going on.
Yeah.
I hope I
very interesting question. Yeah. Thank you very much, Lionel.
Thank you very much. Thank you very much, Lionel. So maybe if there is other question, you can probably respond by mails. And now it's time to let Chris to conclude and speak for the next session of the ESDR Kitchen. Thank you very much. Thank you very much, Lionel.
Thank you, Julien. Thank you, Enikö, and particularly great thanks to Lionel. Our next visit to the kitchen is in 2 weeks' time on the 2nd of June. We're delighted that Lars French, who is chairman of the Department of Dermatology in Munich, will give his personal expedition to searching ingredients of inflammatory skin disease. And we're all looking forward to that. And just to close, as you know, this year's ESDR annual meeting will again be virtual. Last year we didn't have it. This year it's virtual. Hopefully next year it'll be face-to-face. But we encourage you to attend, and abstract submission deadline is the end of this month, 31st of May. Okay, thank you. Hope you enjoyed that, and we'll see you in 2 weeks' time. Okay, bye.